much, Michele. Thank you very much for allowing this manual to be presented to the steering committee EUFORGEN and also broader audience. So this is the ultimate output of the LifeGENMon project, which was a 6.5 years long project from 2014 to 2020 between 6 partners from 3 countries. And we are really very happy that this manual for forest genetic monitoring has come out as an output as wished for, and that it is going to serve for the EUFORGEN community as well as all the other initiatives for forest genetic monitoring and conservation of forest genetic diversity at the national and international schemes. And now I would like to present Marko Bajc, the head of the Forest Genetics Laboratory at the Slovenian Forestry Institute, who was the editor-in-chief and did most parts of, who put most time, thinking and nerves into preparation of this manual. So Marko, please, the floor is yours.
Thank you, Hojka. First, I would like to apologize. There seem to be some technical difficulties on my computer. I cannot seem to figure out why the Zoom keeps complaining about some other application being in the way of my presentation. Hopefully, this will be resolved. Can you see my my presentation?
Just put it on presentation and that's it.
Yeah, it is. Okay. So thank you everybody for participating. I will be presenting the manual for forest genetic monitoring system as designed and prepared within the LifeGENMon project. Now, before we dive into the contents of the manual itself, just a few short words from an editor-in-chief point of view. This, as Hojka has already pointed out, has been a major endeavor, in a way, almost a project within a project. And really countless people put an enormous amount of time and effort into making of this document, which in the end proved to be not just a collection of protocols and findings gathered within the project, but it was also a sort of provided a feedback for the system itself. And as the work on the manual progressed, we also managed to improve and streamline the monitoring system itself. So if we look at the manual itself, it is divided into 10 chapters, which generally follow the sequence of activities that need to be done for the forest genetic monitoring. We are going to skip the chapter 1 introduction, and we are going to start with chapter 2, namely chapter on the plot selection. Within this chapter, we define the criteria for delineation of monitoring regions, which is the first step. So for the species of interest, you first delineate the monitoring regions, the monitoring region. And then, of course, we provide criteria and additional recommendations for the plot selection. Now, within the system we devise, we recommend that at least one plot per species per monitoring region is established, and that each plot contains 50 reproducing trees. Although this number can be, in certain circumstances, reduced, but not lower than 30 reproducing trees. And monitoring region maps, as delineated within the project, are also provided in the manual in the supplementary information section. Next in line is plot establishment and maintenance. Now, in this chapter, we provide detailed descriptions on and the equipment needed for the plot establishment itself. Um, Here we tried to cover different possible scenarios that will be encountered in real life. So we provide protocols for plot establishment for stand forming species, which is the more simple procedure, as well as for species with a more scattered distribution, where the plot establishment is a slightly more complex and complicated matter. And also for establishment of natural regeneration subplots, which is also monitored within the forest genetic monitoring system. Also information and of course instructions on labeling of trees, georeferencing and field measurements that need to be taken at plot establishment, general plot description for which we provide standardized forms which are included in the manual as well as recommendations and instructions on short and long-term plot maintenance and collection of meteorological data. And here are a few pages extracted from the manual itself that show how we deal with this, and we of course provide as many pictorial information and guides as possible. Mm, this is a schematics of how the plot establishment process basically works. So we start with the selection of the wider plot area, which is then followed by the establishment of the plot and choosing the micro location basically first of the plot. And as you can see, there are different approaches depending on the biology characteristics of the species. So for the stand forming species, as well as for the scattered species where more work needs to be done before the establishment of the plot itself in order to identify the locations of the target species within the stand itself. And for the scattered species, we differentiate two basic scenarios. One is for sporadically occurring trees, so trees that are generally found individually within stands, and for the species where individuals grow in smaller groups or clusters. Chapter 4 deals with indicators, verifiers, and background information. So essentially, this is the core, the heart of the monitoring system itself. And here we provide descriptions of the indicators, verifiers, and background information that are assessed, explain their significance for forest genetic monitoring, as well as how everything is tied into the three different levels of forest genetic monitoring that we have defined in order to provide different intensities of monitoring depending on the available resources of the country or entity wishing to start with the forest genetic monitoring itself. If you look at this table from the manual, we defined three indicators that are being assessed, namely selection, genetic variation, and indicator 3 is a combination of the gene flow and mating system. Each comes with their own set of verifiers and background information. And here to the right of the table, you have the levels. And as you can see, if we start with the basic level, which is the most stripped down version of the genetic monitoring, only the indicator selection is being assessed. And for that, not all of the available or defined verifiers and background information. As we move on to the standard level, the number of verifiers and background information that are assessed expands a lot. And the second indicator, genetic variation, is also assessed at the standard level. Whereas at the advanced level, we also introduced the last indicator, gene flow and mating system, and all verifiers and background information are assessed at the advanced level. And of course, logically, the complexity, the intensity of work, and the cost of genetic monitoring increases across the levels from basic to the advanced. Chapter 5 deals with field work. Now, this is the field work that is done with regular periodicity within the forest genetic monitoring. So it does not include the plot establishment because that is just done at the beginning, but all of the regular activities that are done with some described frequency. And two groups of activities are included here. One is the observations and measurements that are done in the field. Here we have both verifiers and background information, including phenological observations and measurements of DBH and height, as well as assessment of mortality survival of both adult and natural regeneration individuals, as well as the abundance of natural regeneration. And the other group of field activities is the sampling. Now, this is only done at the standard and advanced levels, but it involves taking samples for molecular analysis or for seed testing, which is done according to the ISTA protocols. So again, of course, detailed descriptions of how each of these activities is conducted and the frequency at which it should be conducted within the monitoring system is provided. Chapter 6. Now this chapter is probably the one that took the most time and effort to finalize and deals with laboratory and data analysis. And we really worked hard on this one. So if we look at the start with the laboratory analysis, here we provide instructions and recommendations on sample handling, both in the field, in the lab, and long-term storage, seed testing protocols, and of course protocols and recommendations for conducting molecular analysis, which include DNA extraction, PCR, and all of the subsequent steps for both microsatellite and SNPs. Mostly we focus on microsatellites because these were the markers of choice for this project. This mostly has to do with the time at which the application for the project was written, but we also did additionally analyzed SNPs during the project, so we provide information for both. And of course, data analysis is another very important part of this chapter and the entire monitoring system. And here we provide instructions on how to organize your data storage or database. We actually developed our own database for the LifeGENMon project, which is still operational and hopefully will be available to outside users as well in the future. And it provides storage for both molecular data, so microsats or SNPs, as well as additional information such as data from phenological and other field observations. Instructions on how to conduct data integrity checks and data filtering is provided. And of course, the core of the analysis, the calculations of parameters themselves, and finally, the interpretation of values. Now, for the calculations, we really did a lot of work and tried to provide all of the essential equations that are needed to calculate the parameters, so that if a user wishes to implement this in some of their own, in-house developed scripts, such as the R, they can do so without the need to go through countless publications to extract these equations. Another example of how some pages from this chapter look like. Now, throughout the manual, you will find these boxes in which we provide some interesting facts or some tricks and hints how to make your work easier. And here an example, of course, of how to calculate some of the genetic parameters with all of the equations listed. following chapter 6 is chapter 7 on cost assessment. Now, cost assessment was done very, very meticulously, actually, because this was one of the things we tried to consider throughout the project, that we need to define very well what the costs of genetic monitoring are. So we actually did really very detailed cost assessment analysis, and we provide cost assessment from just about every possible angle you can imagine. So in terms of contributions of different monitoring activities, down to the cost per individual verifier and background information, and uh, all of these calculations also provided a sort of a feedback that helped streamline and rationalize really some of the observations that were being done in order to make the entire monitoring system more feasible, really, and more practical to do in the field. In chapter 8, we deal with the decision support for selecting the intensity of forest genetic monitoring. Now, this chapter is aimed at policymakers to help them choose the most suitable level of monitoring according to the questions they need answered and the resources available, both in terms of financial means as well as human resources and expertise available. It is based on a decision matrix which interlinks questions to be answered with levels of monitoring. And of course, taking into consideration the informational value of different parameters as well as their cost of their assessment. And in its conclusion, this chapter provides recommendations on forest management actions that can be taken should some negative changes be detected. Chapter 9 is about guidelines for forest genetic monitoring. Now, guidelines were developed as documents intended for the personnel who will conduct field work. So, Forestry Service and similar institutions. It provides instructions on how to conduct all the field work, including plot establishment and all of the field measurements and observations. And although it is all of these guidelines are included in the manual, they are also available as standalone documents, which are 16 to 20 pages long and can be taken in the field by the foresters. And they were prepared for 7 model species or species complexes, namely the European beech, a complex of silver and King Boris fir, the European black poplar, the common ash, black pine, the wild cherry, and the complex of pedunculate and sessile oak. And to the right, you can see the cover of one of these standalone documents. So in these guidelines, we try to provide as clear information on how to make these assessments and observations. So we also had botanical illustrations drawn particularly for these documents, so that we have really as clear information for the field personnel on how to record all these observations. And of course, in the end, we have some supplementary information in the annex, which includes maps of monitoring regions, all of the plot description and field observation forms, which can be printed and used in the field for recording all the field level verifiers and background information, and some supplementary tables for cost assessment, providing some additional details for those interested. Thank you very much. This concludes my presentation. Here I would like to thank the entire LifeGENMon team and all of the additional people who helped basically completed this project and the manual, and of course also the financiers. Here to the right, you can see a list of contributors to the manual, and you can see it is truly quite long. Thank you.
Thank you very much, Marco, for the very brief and complex presentation. Thank you. Now I would just like once again to thank all the contributors, and that is not just the project beneficiaries and those who have prepared the project proposal, that were mainly Barbara Fussi, Phila Ravanopoulos, Monica Konert, Mariana Westergren, and myself, and also the colleagues from the dissemination part. But it was also the national focal point, the experts from the Transect countries. So from Austria, it was Heino Konrad. From Croatia, it were Davorin Kajba and Mladen Ivankovic. From Bosnia and Herzegovina, Dalibor Baljan. From Serbia, Sasha Orlovic, and helped also by Srđan Stojnic. And then from North Macedonia, Vlad Kondonovski. And also already just before the project started, the steering committee of EUFORGEN has delegated 3 members of the advisory board, and these were Mari Rusanen, Jason Hubert, and Shandor Bordac, who was later on also replaced by Laszlo Nagy, and Shandor has remained as an additional national focal point for Hungary. And also the advisory board also comprised of the representatives from the 3 ministries responsible for forestry, from Slovenia, Alinka Korenjak from Greece, Despina Paitaridou, and from Bavaria, and I never remember the name. So thank you to everyone. It was really a great work and great 6.5 years project. And I hope, I'm certain that this manual will be of well appreciated and used by the international and national community working on conservation of forest genetic resources. Thank you. And now I'd like to ask Michele to lead the discussion and follow up. Thank you.
Thank you very much, Heike. Thank you very much, Marco. First of all, allow me to congratulate well, both of you and all the LifeGENMon team for developing such a remarkable product that I'm really sure that is going to be a landmark in our field for many years ahead. I would like now to open the floor for questions. Please, you can just raise your hand, and then I will try to keep a track on who is asking the floor, and then we can open for questions. While we wait for questions, I can anticipate that we will be making the recording of this session available later on to our channels, so that it will be also be a way to present the publication and also to engage the wider community. Well, Marco, your presentation was very clear. Oh, Sheila, I can see Sheila. Please, Sheila, kindly introduce yourself, and then you can ask.
Hi, everyone. Michaela, you might have known I put my hand up. Yes, so I just got the invitation just a couple of hours ago to attend this webinar from Heike. So I don't work in forest genetic resources, I work in plant genetic resources, and my expertise is in crop wild relative conservation planning mainly. I'm always very interested, although I know very little about, relatively speaking, about forest genetic resources. I'm always very interested in the work that's being done in this field. And I'm participating in the Generous Bridge project as a partner. The university, I represent the University of Birmingham in the UK, in which we're writing this overarching strategy for forest, animal and plant genetic resources. And so I'm interested from that point of view as well. But I always feel like the forest genetic resources community is at least a couple of steps ahead of us. I'm always very impressed with the work that you've done on the development of GCUs, and now to see this manual, I mean, it's absolutely incredible work that you're doing. And I guess we're a bit behind with crop wild relatives, because it was really only in the turn of the century that we even defined what a crop wild relative was. And that we've been working for the last sort of 20 years or so on how to plan and undertake crop wild relative conservation in situ. So to get to my question, excuse me, at the beginning you said that there would be one site per species minimum, and I assume that means at national level. I assume these guidelines are for national implementation. So I was curious why you would only have one site per species, and whether that is considered to be representative, like a representative sample in a country.
Thank you very much, Sheila. I let Hoika or Marco to reply.
Yeah, I think. Hi, thank you for your question. First, I would like to also point out that the discussion section is intended that all LifeGENMon members will try to answer, so not just Hoika or me. So I hope that somebody will follow up. No, the one plot per species is not necessary. Just one. Of course, I think it would be great to have more, to cover, as you said, to cover more. Let's call it ecological niches and so on, where the species is found, particularly in regions where you have, you know, rear edge populations and so on. So yes, it would be great, but we have to understand that all of this is associated with cost. This is why, of course, we said that a minimum of one is enough. That one is enough, where you would, of course, choose the area that you believe represents the most you know, the ecological niche of this species that you are of interest. But certainly, if possible, it would be great to have more.
Okay, thank you very much, Marco. I don't know if Hoika or any other of the contributors of this project would like to add anything else. Otherwise, we move to the next question.
I may add something if possible.
Please, Phil, go ahead.
Yes, so this choice is made under the premises that there are more units, of course, in its country. So it's not that one unit is going to be in a country, is that there are many more units, but one is chosen for genetic monitoring, likely one that is anticipated to face problems with climatic change or other anthropogenic pressure. So if in the unit that faces the most challenges, we find out that there is no serious threats in terms of the genetic monitoring assessment, that would likely hold also true for other units that are under less stress. So ideally, as Marco said, it would be nice to be more, but it's always under the premises that there are many more units anyway, conservation units, and that we focus on a unit that is likely going to face problems. Thanks.
Thank you very much, Phil. Thank you very much for pointing out and clarifying. I see now Mari. Please, Mari, go ahead and kindly introduce yourself.
Thank you, Michele. So I'm Mari Rusanen from Finland, National Coordinator for our Forest Genetic Resources Program, and I was also had the right to follow very closely this project from the advisory board. So I just wanted to continue on the previous question and point out two very, very obvious things. And one is the cost pressure, and typically at national level, I would need to make a decision if I have one plot for 2 species or 2 plots for 1 species. And I know that in many other countries where the biodiversity is higher, the pressure for covering all species will be much more higher. And then the other thing I would like to point out is that obviously this value of the single plot in a country increases enormously if we manage to get a pan-European cooperation and a network of these plots. Thank you.
Thank you very much, Mari.
If I may, thank you, Mari. That's exactly.
Please go ahead, Hoika.
Yes, that is exactly the point, and that is why we have to start with prepare these big forest genetic monitoring regions with the help of all these experts, national focal points from the translate countries. And the main point would be that there is, okay, at least one plot per bigger monitoring region, if possible, 1 per country. However, in Slovenia, for example, we have 75 forestry species. In Serbia, they have around 300. I believe it might be a little bit difficult to have everything done in 1 country. And that is indeed why this manual should be as widespread and hopefully accepted as possible, and that EU4GEN accepts, contributes in its in the long-term for genetic resources strategy, that monitoring is promoted through the community to be implemented at the national level widely. Although we would also hope that some kind of international support would be added to these national efforts. International support, I mean financial. Michele?
Yes, thank you very much.
Michele, could I add something on this?
Phil?
Of course, please.
Yes.
Phil, go ahead.
First, thanks, Mari, a very interesting point. And further to what Hoika had said, of course, it's impossible to have monitoring units in every country for every species. But if we manage to have, let's say, a network of around 10 units at the European scale for the most ecological and economically important species, that would be, at least in my view, a very good basis to assess the maintenance of genetic variation over time. So not every country would have all the units for all the species, but if we have for these species 10 units at the European scale, which we monitor, it's a good basis to start evaluating the change in the genetic diversity over time. Thanks.
Thank you very much, Phil. And I don't know if anyone else wants to add something. Otherwise, we can go to the next question if there are any other questions. Sheila, again, please go ahead.
So is there already a commitment of the countries to undertake this genetic monitoring, to put the resources into this? sorry, I don't know anything about the kind of that sort of
sorry, I
kind of
didn't get your
political
question.
the policy background or anything. I don't know anything about it. So.
Regarding the policy background at the national level, that was the question.
I guess so, yeah. So whether there is
Yep.
already a commitment to
Yep,
undertake
yep.
this genetic
Well,
monitoring.
within the project, we had one implementation action, which was building these background documents for the partner countries and for the transact countries, hopefully, to propose the development or modification of some national rules which would support also the implementation of forest genetic monitoring at the national level. In Slovenia, we have included the need for conservation of forest genetic diversity and consequently of the monitoring of changes of genetic diversity into the regulation on forest management and wildlife planning. And it is also included in the guidelines about how to prepare the 10-year regional forest management plans, which are under changing just now. So they started in November last year and they're going on. These 10 years regional forest management plans, they're going on this year. In the other partner countries, so there is a permanent discussion between the Teisendorf Institution for Genetic Studies with the Bavarian Ministry. And in Greece, we also had, so in Slovenia, we also had a partner from the Slovenia Forest Service as within the LifeGENMon team. And in Greece also they have, there is a full partner, the decentralized part of the Hellenic Ministry for Agriculture for how is it called? Fotis?
Decentralized Administration of Macedonia and Thrace, it's the original name, official name. Yes. And we are dealing with the forestry and the agricultural matters and affairs. And we have also tried to, and we are in full discussions how we can integrate forest genetic monitoring to the forest management plans with specific regulation and specific legislation.
Mm-hmm. So this is this was already this implementation at the national level. It was already the part of the implementation actions throughout the LifeGENMon project, and it is still also part of the afterlife communication plan just now. In order to implement the forest genetic monitoring also in the transact countries, we have also, during the project, organized 5 workshops for forestry practices and forest policy or decision makers in Jastrebarsko in Croatia, in Sarajevo, and then later on also in Banja Luka in Bosnia and Herzegovina, and in Novi Sad for Serbia, and in Skopje. And for during the workshop in Skopje, there were also representatives not just for North Macedonia, but also from the lower part, from the Kosovo community and from Bulgaria. So all these countries were checked, our national focal points, the experts, they have provided the overview of forests, forest management planning, forest reproductive material, and the possibility of conservation of forest genetic resources, including forest genetic monitoring in their countries. And we have already presented them the background of the forest genetic resources programs at the national level, for example, C4GEN in Slovenia and of the manual for forest genetic monitoring. The problems in these different countries are rather different and rather difficult. And during the workshops, we have not only concentrated on promotion of the monitoring itself, but also on conservation of forest genetic resources through practice. It means mainly a bit on adaptive forest management, but mainly on the certification system of forest reproductive material, which is used in reforestation, replanting, and so on. And also because forest genetic resources do not know borders, and we are interested in increasing genetic diversity by testing provenances from further southeast to further to the northwest. We were also considering the possibility of this testing and transfer of forest reproductive material across borders in the future. For the other countries, especially for EU member states, it's up to EUFORGEN and also the other technical working groups on forest reproductive material, for example, to define the European strategy for conservation of forest genetic resources and to include the knowledge, for example, into the activities, presentations, discussions, for example, of the Standing Forestry Committee at the EC level and into these overall strategies.
Okay, thank you very much, Hoika. Thank you very much for the clarification. I see AWG want to take the floor for another question. I will ask you to please present yourself.
I would like to add to what Hoika said.
Barbara Fussi
from Heissendorf. Just to add a few comments and from our experience, because in Bavaria, we will continue with the observations and what we have experienced, we want to continue. And we had in Germany a project where we implemented or where we established plots for spruce and beech. And what really helped here, and most of the countries and most of the partners here will also continue. And what really helped and what would really help to implement, I think, would be like for the countries who have no plots yet, to start with a project to implement the plots, to start with the basic analysis, with genetic analysis. Because then it would be much easier for the countries to continue with the observations. But if there is no like starting finance, I think it would be much more difficult for each country to start with the observations and with the system for genetic monitoring.
Mm-hmm.
Thank you very much, Barbara, for your qualification. I don't know, Hoika, I see you want to react directly, or otherwise I can ask if there are any other question. Please, Hoika, go ahead.
Michele, in fact, I was trying to press on you because the point is that at the national level, it depends definitely on the, on the funds, on the budget, on the level of the importance of forests and forestry at the national level. This is also, for example, why our general dissemination was aimed at the importance of presenting the importance of forests to the general public from the children onwards. But anyway, at the national level, it might be in some countries very difficult to go for strict directly for forest genetic monitoring, and that is why it would be really useful if Euphorgen, for example, would propose, support, and also the other existing Horizon projects, such as Generous Bridge or others, would propose to the EC different DGs, or maybe just strictly to DG, either agriculture, environment, or climate, to support such the funding, some kind of baseline funding for maintenance of GCUs and monitoring of genetic diversity in them. Michele, I think.
Yes, thank you very much, Hoika. Thank you very much. Sufficient funding.
Yeah.
Like Mari was also indicating and sharing in the chat, the funding that we currently have in Europe is hardly sufficient to ensure the conservation activities that we have already committed. So like you say, getting sufficient funding would be something really important. And we will make sure that we will develop a proper framework so that we can present this issue to funding agency. Now, before closing, I would just want to ask if any other of the contributor of this important manual want to add anything. Otherwise, I will ask Hoika to wrap up and close this webinar. So last call for any additional contribution. Wish to express position or view from other contributors of this manual.
Mariana, Phil.
Then Hoika, I can ask
Barbara,
you to
Darius,
closing remarks, and then we can thank everyone. Please, Hoika, go ahead.
Mladen has put the hand up.
Yeah. Yes. Just through a few words, it was really very visible from that book that it was done a lot of and big job was done during this project. And I'm really glad from my point of view that I was a little part in that, but really you done great job, and everything is really visible from this presentation, but much more from the book. That's all from me.
Thank you, Mladen. I think Sasha Orlovich is also present.
Yes, hello.
Hello, Sasha.
I have nothing to add. I'm very satisfied that we really have an excellent document, and I'm pretty sure that we will apply it in Serbia.
Excellent, perfect. Thank you very much. I hope in whole Serbia or just Vojvodina or some other parts.
In whole Serbia.
Okay.
Hoika.
Yes.
Just one small comment, if I may, because I'm reading the chat. As Marco presented, the basic level of monitoring is not that expensive. So it's not that a big, so to say, difficulty to find this type of funding support. But what is important, and apparently this is the point that Barbara raised, is the establishment of the plot, to get the thing off the ground. So I think it's a little bit different story to have the funding to establish a monitoring plot, and then to have the funding to maintain at least the basic level of monitoring. So I guess most of our efforts at this point would be directed towards finding the funds to establish plots. And then as far as the basic level goes, I think things are not that much hard. Of course, if you try to do standard level or advanced level, then you definitely need the funding that has to come from some source. But the establishment, I think, is the main issue today. Thanks.
Thank you very much, Phil. Michele?
Yes, Hoika.
And would you like to stress anything else, or I just finished?
Well, I think we can just conclude. And well, I want to thank all the contributors of this manual, and especially Marco and yourself for bringing it up and raising both awareness and also coordinating the development of such a major effort. And I think I would like to conclude taking some of the word that Phil was sharing with us, that what is really important is that starting the effort. And some of these activities can be really demanding, and something can be really pricey, can be really expensive, but with the minimum effort. But we really need to have a commitment of countries to start with the baseline, to start defining this monitoring process that then can be follow up throughout the years. So that's, I will just conclude thanking everyone. I don't know, Heike, you want to say something else? Otherwise, we can close and thank everyone.
Yes, I would just like to thank you again to the whole team, which was really great. Everyone responsible for its obligations and leading them and contributing to the actions. But especially really, and also all the national focal points, the experts, the steering committee members, and so the advisory board members, and so on. But especially, I would like to thank Marco, because he really needed good nerves to put everything together and to, you know, we had innumerable meetings, even twice per week for the last year. In order to put this manual together, and it's not only excellent regarding all the scientific part, all the details, but even at the end also the design. Also, I think all the drawings which were done by the professional illustrators organized by Katja Sonnenschein. They were also did their very good work, and this part of activities, it also needed lots and lots of interactions by the different team members, by, for example, Marco and Rok, and then also Andrej Breznikar and other colleagues from our institute, from the Slovenia Forest Service, from Teisendorf, and from both partners from Greece. And so really this was an exceptional work, an exceptional team, and an exceptional chief editor. So I would like to thank everyone again. And I hope the output will be widely used and implemented internationally. Thank you.
Thank you very much again. Thank you for everyone for attending. Have a nice week ahead. Thank you, Heike. Thank you. Bye.
Thank you.
Bye-bye.
Thank you. Bye-bye.
Bye.
Bye.